Hey, here we are. I tried streaming this earlier today, and the internet just wasn't playing nicely, and I tried to do it in Teams, and I forgot to click record. But here we are. So we're going to try a quick lesson. I just thought it'd be kind of neat to talk about how it is that we can test for COVID-19. You hear a lot about testing. You hear that they were slow to get testing in the United States, and that we have it much faster here now. But we'll talk about how it is that we actually test for that, because it's kind of a neat thing, and it relates to what we've been learning. So I spent some time today actually looking up the guidelines for COVID-19 testing, and it's kind of neat because one of the things you discover is that it uses some pretty simple lab techniques, things that aren't all that different from what we've done, and then some fairly sophisticated lab techniques. So let's talk about that. So the first thing to understand is that when they test for COVID-19, the first thing they have to do is they have to collect a sample. And in the guidelines for testing that we're using in Ontario, there's two different kits they can use to collect a sample, but they have to be collected in a swab that can then be held safely. So if you imagine we've got, the nasal cavity of an individual here, it is currently collected using a swab like this. And that swab is inserted nasally, and they take a swab of the nasal cavity. So if you are to get COVID testing or SARS-CoV-2 testing, coronavirus testing, whatever you want to call it, they're probably going to do a nasal swab. They can also do a throat swab, but from what I've read, that is not the preferred mechanism. Then they're going to take that swab and they're going to seal it, and they're going to ship it off on ice. So it has to be sealed, cooled, and sent to the lab. And once it's been sealed and cooled and sent to the lab, then it's going to have to be processed in that lab. And when it's processed in the lab, I found some conflicting reports, and I'm not sure exactly which of these two methods are used, but it's the same basic idea. At a bare minimum, they're going to take a sample tube, and that sample tube is going to have some liquid in it like this, and they are going to put that swab that they collected into that sample tube and stir it around. And that will dissolve the material that they swabbed into the solution. Now, what are they doing when they do this? Because they are trying to find out whether or not you have the virus inside of you. And so the question then is, well, what are they really looking for? And this is where there's a little bit of unclarity for me. From what I read, they may at this point centrifuge it. And if you're familiar with the centrifuge, we actually have one in our lab that I've used at various points. But you can put a bunch of sample tubes into it, like this. It's just a device that looks kind of like that. And you would put your tubes in there, and we would put this particular sample of ours here. And this device then spins really fast. It's called a centrifuge. So I've seen it suggested that it is centrifuged. I've also seen other cases where it's not, and where they just work with it as collected up here. I'm not entirely sure which, but I will look into that a little further. But this is a possible step where you centrifuge to separate into layers. When you centrifuge something, it separates it by weight. And what they're looking for, specifically, what we want to test, is we want to look at that tube. And now if we had centrifuged it, it will kind of clear into layers of things of different weight. And what we want is the sample that contains viral RNA. And this wouldn't just be viral RNA. Really, it would be RNA/DNA. We haven't talked yet about the fact that, while humans have DNA, viruses have RNA. And it might do well to kind of briefly cover what the difference of those is. But for the moment, I'm just going to explain it as if viruses had DNA. And at the very end, I'll talk to you about that. So this is just getting the sample ready to test. So we've collected a sample. Once we've collected that sample, we've processed it. And now we're actually ready to test it in the lab. So the actual genetic testing. And in this case, we're going to use a process called PCR. And PCR is really interesting, so we're going to talk about it because it relates to actually a lot of stuff that you guys have learned in biology class, which I think is kind of cool. So PCR is called polymerase chain reaction. And it's a laboratory process that can do a lot of different things. But one of the main things we use it for is to identify whether or not DNA, a certain genetic code, is present in a sample. So if you know in DNA, as we've talked about, it's double-stranded. It's that famous Watson and Crick discovered double helix shape that looks kind of like this, right? And that's the worst drawing of PCR or of DNA that anyone's ever done, but that's okay. And we could look at a particular segment of that DNA. Maybe we could look at this chunk right here. And if we looked at that really closely, we would see that it looks like a bunch of nucleotides together. Now remember, DNA itself actually has a phosphate and a sugar, and those make up the spines or the chains, and then the actual coding information is on those nucleotides. And so if we looked at this, we would actually see like A, G, C, C, G, T, C, A, ... And it would keep going in both directions. And then there's a complementary strand. Remember, one of the things that makes DNA interesting is this redundancy pattern. So the T lines up with A, C with G, G with C, G with C, C with G. So guanine, cytosine, tyrosine, and adenine. And they always pair up like that. So that is the actual DNA we're looking for. Now, PCR can identify and amplify, and we'll talk about what that means in a minute. What's it mean to amplify? But it can identify and amplify specific codes within a DNA sample. So we could look for a specific sample. So no different than if I wanted to right now, I could press Control F in here and look for the word chain, and it would look through all the words on this page and find the word chain and highlight it for me. PCR is a physical mechanism we have that actually lets us do that. And I'm going to show you that, by the way. I'm going to press Control F for find here. Oh, that's not how you do it in this. Now I'm just being stubborn. But if I went here to view, oh, there's the search button there. And I could search in my current notebook. In fact, on my current page, I could search for the word chain. Oh, I can't read my handwriting. Or maybe it just hasn't had a chance to do so yet. But, just like I can search for a word here, and it will look through all of my notes and find it. See, and here they all are, all the different notes that have that word. PCR serves a similar function with your actual genetic code. So that was a really ineffective thing. And if I were like an effective teacher being careful here, we could do that. So if we actually looked at the genetic code, we might be like, okay, there's a G and an A and a T, and I don't care about any of this. But then all of a sudden we get into the important part of the DNA here. There's this C, G, C, T, A, C, and then blah. This could go on for a long time. It could be a really long genetic code. And then maybe it ends A, T, C, G, C, T, A. And then again, we've got just kind of, after that, a whole section we don't really care about. Now, of course, this is going to be paired with a complementary strand, because DNA always has a complementary strand. Here I'm showing it in green. So I'd go G, C, G, A, T, G. You can tell this isn't actually random because I've been really afraid to have repeats here. Truly random data would almost certainly have some repeats. It's a good reminder that we're not very good at making things actually random, but that's okay. And so this DNA would stretch on long, because DNA code's really long. But if this section here was the section I'm interested in, then we can use PCR to find that section of DNA, which is pretty cool. And so the first things that have to be done is that they have to figure out what section of DNA they want to look at. And fortunately, they've been able to do that because they have looked at COVID-19. They've looked at specifically the SARS-CoV-2, this novel coronavirus that contains the actual genetic information we want, and that data is available. They know what the code is, and I'm going to show it to you later. I had it up earlier, and I haven't pulled it back up since, but I will pull it back up so I can actually show you the really specific coding sections they're looking for. It's really neat because they know precisely the code just like this that they actually want to find, which is pretty cool. So now PCR itself is a 3-step process. So polymerase chain reaction testing is a 3-step process. So basically, you would take your test tube, and this has your sample that includes the DNA you want to test in it. So this is your sample with DNA to test. And we're going to add a few things into it. We're going to add, first we're going to add some like salts and buffers that just help the conditions for this to be right. So these are just conditioning. They just make sure that the water is the right pH, it's the right salinity, it's things like that. They don't really do the important part. We're going to add a bunch of what we call primers, and we'll talk about what those are in a minute. And then we're going to add another solution that's going to actually contain polymerase and the nucleotides we want. So we'll put, oh gosh, didn't want to do that, now did I? Oh, there we go. Okay. So then we're going to add polymerase and nucleotides. And this is just to get the solution ready. And we'll talk about what each of these things does, so don't worry about it. But this would be the lab step. If you're actually in the lab, this is what we would do. Now, our first step, just like when we replicate DNA, our first step is to unzip the DNA. And in the cell, this is handled by an enzyme. I want to say it's called spiralase or something like that. But it's an enzyme that inside the cell does this for you. But we don't, we're not doing this, the way an organism does it. So we have to figure out some way to unzip the DNA, to separate the two complementary strands. In other words, it starts out like this, with one helix like that, and the second strand following along it like that. And we need to figure out some way to take that and make it look like this. So we have access to both strands. And it turns out that's actually not that difficult to do. We just need to heat it. So we take the solution and we heat it to about 95 degrees Celsius, which is pretty hot. That's almost boiling. And this results in a process called denaturing, or denaturation. But it just means that it is unspiralized. It's unzipped. It's ready to work with. Again, your cells don't do this when they have to replicate by heating up to 95 degrees. They have a different method. But for this process, for the PCR process, we unzip the DNA just using heat. And this doesn't take very long. It's pretty quick, and that happens. And then the second step is primers attach. So we said that when we set this solution up, we attached primer, or we dissolved primers into that solution as well. And primers are something you pre-make. So they are designed in a lab and pre-made. So these are made in a lab. To find specific DNA code. So they are pre-made little segments of DNA that are ready to bind. So for example, if I had a primer whose code was, I don't know, ACGC. Now this is too short. A real primer would be longer, but that's okay. This would be ready to bind to what? Well, it'd be ready to bind to TGCG. Does that make sense? So we know that how complementary DNA works. So we put little snippets of DNA that we've pre-made in the lab, and they are set up so that these are going to find this and stick to it and bind to it. And what you do is, if you know the segment of DNA that you're interested in, so in the previous step, we separated these. And if in this previous step, we were really interested in this section of the DNA there, well, now it's been split into its parts here and there, if that's the part we're interested in. Well, now, if we come down here, those are the segments we're interested in. The primer's job is to mark the endpoints. basically the start point and the end point. If it's a really short segment of DNA they're looking for, they can actually design this primer to be just the length of the whole section they're looking for. But you can't let this get too long or it will start to fold up on itself. So the primers have to be fairly short. So most often, it's carefully designed to mark an in point and to mark an out point. So you would use not one primer, but two. One to mark the in point and one to mark the out point of the section you're interested in. And there's interesting videos on how these primers are made too. We'll use different primers to mark the in point and the out point. And we'll talk about why we mark one on one strand and one on the other, but of the section of DNA we're interested in. So as a rule, we don't make primers to cover the whole thing, or we don't make PCR that covers the whole thing exactly. It finds the opening point and the closing point. And so we can show what this looks like with our actual example here. So we made up this made-up example here. So I'm going to take this. I'm going to copy it. I have the technology. So we'll copy that. And we'll go down here and we'll say, if this were the section we were actually looking at, well, in the previous step where we got it really hot, up to 95 Celsius, we would have taken it and actually split it into two separate strands. Just like this. Now, we would have a primer whose code was, say, GCGA. Now, again, it would actually be longer than this, but that's okay. We're just going to simplify it and say that. And that primer is going to find this section of DNA, and it's not like it's looking for it. It's not that this primer is smart or programmed or alive or anything like that. It's just that it fits well along this. So as long as we've put a lot of primer in there, these should bump into each other. And when this bumps into that, it's going to bind because it's a match. It fits. And we'd have put another primer in that looks at the end of this segment we want. And so this would be a GCTA. And again, the real thing would be a little longer than this, but the principle holds, and it would lock in there. And now there's a primer on each of these strands of DNA that is ready, well, for what? Well, what we're ready for is cooling back down and allowing this third step to happen. And the third step is cooler temp, and it is polymerization. And this is where we add something called DNA polymerase. In fact, specifically, we use Taq polymerase. And that Taq refers to an organism which is a thermophile. Because it needs to be able to work at high temperatures. Because even though we say this is cooler, it's still happening in the 50-degree range. Or maybe this one is up at 70. I'll look up the specific temperatures at the end. But this is at a higher temperature than normal DNA replication would happen. So we need from a thermophile. And obviously we've already done this. This also requires the actual nucleotides. So the actual A, G, C, and T nucleotides. Because if you haven't added those, the polymerase has nothing to add on. But the polymerase is the real thing that normally in your cells fits the A's and the G's and the C's and the T's onto the template DNA to make complementary strands. So this makes complementary strands of DNA. And so it's actually going to take the sections that were ready and waiting, and it's going to fill in the DNA. Because if you look, basically, now that we've cooled down a bit, the DNA is kind of able to refold. But this section where the primers is, they'll be held away from each other and kept from refolding. So there's a primer there, and there's a primer here marking the in point and the out point. And what's going to happen now is DNA polymerase will come in, and it will make this complementary strand. And it will make this complementary strand. Just like that. And if we look at that like this, if we take this example again, we can see what's actually going to happen is that DNA polymerase, again, this molecule that just always exists in living organisms, is designed to replicate the DNA. We didn't design it, it just already existed in nature. So it's going to come in here, and it's going to fill in all of the missing information. So this will go T, G, and it would fill in, it would match all these in the big stretch, and then T, A, G, C, G, A, T, and it probably will keep going for a little bit here until it kind of runs up to where it doesn't fit, because it doesn't know where to stop on this strand. And that's okay. We'll explain later on why that doesn't really make a difference here, but it's going to replicate in this direction, away from the primer, and just keep going. And this one's going to replicate away from the primer in this direction. And so that'll be a C, and a T, and an A, and it'll fill in this gap all the way here, and then a C, and an A, and a T, and a C, and a G, and a C, and a T, and an A, and a G. And these have to go in opposite directions for a very specific reason, which is that they always go from what we call the 3' to the 5' end, which just has to do which side of this is the sugar side of the spine and which side is the phosphate side of the spine, which we can explain at a later time. But so now we have 2 copies. So when this is done, we've duplicated just the DNA we're interested in. Boy, this is really acting up right now. Sorry, give me a minute there. Oh, and OneNote crashed. I've been asking a lot of my poor computer today. All right, so let's try that again. We now have 2 copies of the DNA of interest. But note, we don't have 2 copies of all the DNA on this thing. We haven't replicated all the DNA. All we've replicated is this one little section that we were originally interested in. Just that middle little section between the start primer and the end primer. And now, really comes step 4. And I know that was just 3 steps, but that because this is just repeat. So we can heat and cool over and over again. to repeat this process. And that isn't nearly as complicated as you might think. This is actually, once it's all set up, this is relatively the easy thing. So if we looked at last time, we had that original green, that original orange, and these actually in the end up stuck together again. But in that middle section, we have kind of this strand that I drew in gray, which is now a copy of each. Well, what can happen now is we can heat it up again, and in so doing, we get the green, and that gray section, and that gray section, and that orange section, just like that. And then, when we add the polymerase, yeah, I'll just be a couple more minutes. Almost done. When we add these sections here, and we add the polymerase, now it'll duplicate that, and duplicate that, and duplicate that, and duplicate that, from primer to primer. Right, and remember, it did that by adding primer here and a primer there, and a primer here and a primer there. And now we have 4 copies. And if we do it again, we would get 8. So the first round we get 2, then 4, then 8, then 16, then 32 copies. And the trick is, we just keep doing this until we have millions or billions. And it doesn't take as long as you might think. Because all we need to do is keep in mind that every time we're multiplying by 2, and 2 to the exponent 30 is over a billion. So assuming everything's worked perfectly, and of course it doesn't work perfectly, but if it did, if you did this just 30 times, you'd have over a billion. In practice, for the COVID test, they do it, I believe, 42 times is the specific thing I read on the guidelines for this, which is a lot, but the whole thing takes not that long. It takes on the order of, just a few minutes. Now, they actually, when they test for coronavirus, they do something really clever, which is to make life a little easier. They just kind of... they kind of repeat over and again. Now, once they have the sample, how do you test it? How do you actually see whether you've got a bunch of copies of this or not? Because if you don't have the coronavirus, this DNA doesn't exist, and the primers have nothing to latch on to, and so the polymerase does nothing. If you don't have this DNA, if you don't have the section it's looking for, nothing happens. But if you do have the DNA they're looking for, it makes billions of copies, which is kind of cool. So once you've done all this, how do you know? Well, there's a number of ways to test this. And there's a few ways we can make this visible. Something called fluorescence, where they can actually make the primer so that in addition to having that DNA on it, it's got a little piece of protein that when exposed to UV light will glow. but only after it's been removed from the primer because the primer's bound. That's one way. They can do spectroscopy that just sees how much DNA total is in the solution. And this can actually even be done in real time. In fact, for coronavirus testing, it is done in real time, where as they're doing it, they're running it through spectroscopy, and they can see how much fluorescence is happening over time and see that it goes up. And this is fairly quick. Just to give you a sense, for the current COVID-19 test, this is the cycle they're using. At 55 minutes for 10 minutes for reverse transcription, we'll talk about that in a minute, and then followed by 95 degrees Celsius for 3 minutes, which is the unfolding. The unfolding only takes 3 minutes the first time, and then 45 times in a row, they heat it up to 95 for unfolding, and then drop it to 58 to allow the primers to bind and the replication to happen. And then back to 95, then back to 58, then back to 95, and back to 58. And they do that 45 times. And then they're done. So I've got this kind of diagram here that shows it, and I think shows it pretty well, actually. And you can see that all they have to do is first they have to get it hot to denature and add the primers, and then they cool it to allow polymerization to happen. And then again, you heat it to allow it to denature and the primers to anneal, and then you cool it for polymerization to occur. And that's it. That's the process. Now, there's one more thing to think about. And it's not a big deal, but is of note. It's worth mentioning. Which is this virus, SARS-CoV-2, has RNA, not DNA. Now, we haven't talked much about RNA yet. We were going to, and then class was rudely interrupted by a global pandemic. But RNA is a simpler form of genetic material than DNA. It's called ribonucleic acid because its sugar is a simple ribose instead of deoxyribose. This is similar to DNA, but it is a few major differences. The biggest one, the most important difference, is that single-stranded. So instead of having that automatic backup and protection of the complementary strand, the nucleotides in this just make a single strand. So you get the kind of strand like that, and then the A, and the G, and the A again, and then C, and then G. So there's that. And then secondly, instead of thiamine, RNA has a different nucleotide called uracil, which we show as a U. Not a big difference, it just functions like thymine, but it is a different compound. So this may sound like it's a game changer, but it actually just adds one step. And the reason it only has to add one more step is because nature already has a protein that allows you to take, an enzyme that allows you to take RNA and use it with DNA and sort of convert it to DNA. Just like you might convert a file on your computer from one format to another, your cells are able to convert DNA information into RNA information. That's the way it usually goes, actually. But it's also able to take RNA information and convert it into DNA. So the RNA must be given a complementary strand of DNA. And it uses an enzyme called, and again, this isn't something we've made, it's a naturally occurring thing in cells because they do this conversion all the time, called reverse transcriptase. And it's called reverse transcriptase because the more common conversion is converting DNA into RNA. And if you take grade 12 chemistry, you'll learn why that is. Transcriptase. Okay, I even spelled that right. So, if we have here our viral RNA, and because it's RNA, it'd be CCACGU, C, and then here's the section we're actually interested in. AUUCGGAUUCGA. I'm going to do this just because I'm going to put some dots there, and we'll say CGAU, and then we'll say, okay, UAC. All right. So that's the viral RNA that we're interested in. The first thing we do is add a complementary DNA strand by adding nucleotides and reverse transcriptase. And again, the amazing thing is we don't have to design something in the lab to do this because nature already does it for us. So it's going to make a complementary strand, GGT, because now this is DNA, not RNA, GCAG, and now here in the complementary strand in the section we're interested in, no U's, TAAGCCT ... GCTA, and then again ATG. And again, the colors aren't because there's something special there, but rather this is the section we're interested in. And now you can just do PCR as normal. So the hardest part about PCR is not doing it. This is actually a fairly simple process. It requires some specialized equipment that you have to own in your lab. It's fairly expensive, but not incredibly so. And once the lab is set up, the actual doing of it is quite simple. The hardest part is designing those primers, is figuring out what the code of DNA you're going to look for, and then looking for it. PCR actually has a lot of other uses. It's not just for detecting if DNA is present, though that's one of its most common uses. And the other thing is the COVID test actually does 3 levels of PCR. To make it quicker, they do a first quick one that has a primer code that isn't all that complicated, so this can be done fairly quickly. But it will actually have a lot of different kind of false positives, which is to say there's a lot of different viral DNA codes that could set it off. It isn't entirely specific just for the current SARS. So if this is positive, then they give a more specific test that has fewer false positives. And if that's positive, then they have a really specific test. And the idea with all 3 of these is both to mean that you don't have to do as complicated a test if somebody shows negative first, but also to have some fail-safe there. So that's it. There's a lot more interesting things to know about PCR, and I'm going to throw some links up on this lesson as well. But I just thought it'd be kind of fun to cover some of this material. Yeah, so that's that. If there's something that interests you, if you have questions about this or other biology, if you can't tell, I'm kind of bored and kind of jonesing to teach something. So please feel free to send me a question, and I'd be happy to try to answer it. And I'd be happy to try to come up with some material. And if you're finding this totally useless and you're like, this really sucked and I could have just found this out on the news, but I'd really rather do this other thing, well, tell me that too, because I don't want this to be a waste of your time. This isn't testable material. You don't have to know this. I'm just trying to kind of keep your head in the game a little bit, keep you thinking about things, and keep myself from going crazy. I've spent most of my time building, this is part of what will be a face mask. So I have some plexiglass in my shop that I've cut to the form of face shields, and this can go in front of a medical practitioner and keep them protected from bodily fluids and from airwaves and things like that. Hospitals have been requesting that people who have 3D printers make these, and I was able, before non-essential businesses shut down, to get to Hamilton and buy some sheets of plexiglass, thin little stuff. 30 thousandths of an inch thick. So I've been doing that, and I've been, going on lots of walks while avoiding people. But I'm a little bored, and I'm sure you are too. So hopefully there's some help to you. Stay safe. Call a friend, call a family member, call a loved one, check in on them, make sure they know that you care and that you love them. But the most important thing you can be doing right now is just staying away from other people, washing your hands regularly, and staying safe. All right. I hope to see you again soon. Ciao.